So it's now our final speaker, and it's my pleasure to introduce Professor Al Darby from the University of Liverpool, who's going to continue our surveillance theme and talk about sequencing and surveillance in the context of clinical and wastewater. Thank you very much.
Thank you for the invitation here today. I am not going to do a very good job in hiding my agenda. I'm a professor of genomics at the University of Liverpool, and I'm going to talk to you about genomic surveillance. So who can guess the agenda? No, but I do think that one of the things that we've learned recently is just the power of genomics in understanding outbreaks. And so what I want to talk to you today about is introduce myself very briefly, talk a little bit about genomics and genomics technologies, and then go on to talk about pandemic genomics and surveillance and diagnostics. And a lot of what I want to talk to you today is based on experience through being one of the co-directors of the Centre for Genomic Research. I'm currently an advisor to PathSafe on tracking foodborne infections, and I work closely with NHS colleagues. But formerly, I was one of the deputy directors of COVID-19 Genomics Consortium in the UK and a subject matter expert for the Rosalind Franklin Laboratories, honorary contract with the UK HSA, and was part of the UK HSA Genomics Technical Working Group. So I normally give this talk to a slightly more mixed audience, and so one of the first things I always do is make sure everybody knows what I mean when I talk about genomics. And from this point of view, for you today, really what I want to talk about genomics as being is an end-to-end process. It starts at a design of an experiment or surveillance, all the way through to getting that data analysis. So genomics technologies allow us to look at a whole wide range of things, from genomic structure to individual SNPs in a gene, look at protein coding, look at RNA expression, and of course epigenetics, ways in which those nucleotides might be changed. So genomics quite often provides these days a foundation for downstream studies, and having a genome is quite often the first place where people start, or one of the first questions that people ask. The reason this has been able to happen is because, and you will have seen this slide before, is because of the massive decrease in the cost of sequencing. Now, one of the things I want to say to you today is that that has leveled off. Okay, and that is no longer the case. We're going through this exponential phase of decrease in sequencing costs that we were seeing about 10 years ago. Things have leveled off, and we're up to the point now where it costs most people about £20 to do an E. coli genome, and you can do a human genome for sub £1,000 now to sort of £400, depending upon the technology that you use. Now, the reason I tell you this is that quite often people think that cost is a barrier to doing genomics. And I think it's important that we have a dialogue about how to reduce the costs and how to give people access to those reduced costs. Because the first human genome cost the same amount as putting a person on the moon at the time. And so we're in the situation where we can do a genome for £1,000. So I'd like to think that that would mean if we could go to the moon for £1,000, that's where we'd be spending our holidays, okay? Or at least we'd go once. So I think price as a barrier shouldn't be an excuse. If it costs too much, then ask if you can do it cheaper, and we can always try. So technology's been a great part of this, enabling us to do this work at higher throughput and reduce cost. So things like the Illumina sequencing, which you come across, is a very unique piece of technology. It uses a special type of PCR to amplify the DNA onto that chip and then sequence it in very small pieces. The advantage of this is that you can get large amounts of data, and I mean large amounts of data. You won't be able to store it on your laptop, but also it gives us very accurate sequencing, very trustworthy, very reliable. We've also begun to have single molecule sequences available to us, which give us new insights. So instead of looking at these clusters of sequence that we've amplified, we can start to look at the individual molecules and quantify those and look at those independently. So things like the PacBio, the little picture there is of a little tiny reaction well, and at the bottom of that is tethered one single polymerase, and then we put one single molecule of DNA around that polymerase and let it sequence itself, and then watch it in real time as those bases are incorporated to see how the sequence is being synthesized. Okay, really cool bit of kit, really helps us with doing things like de novo sequencing, where we want to build genomes really accurately, and gives us really excellent data. Another one of these technologies is the Oxford Nanopore sequencer, which you've heard about before, and this again is a very, very different type of technology, where the DNA or the nucleotide is now going through that pore and disrupting the ionic flow through that pore. And through the magic of AI, we can tell which bases are currently in that pore and being sequenced at any one time. So again, this is a very fast technology, and is actually plagued a little bit by the fact that it's also slightly more noisy and inaccurate. But one of the reasons why you're hearing a lot about it is that I can bring it to a conference with me as a fidget toy. Julian's laughing. So this is an Oxford Nanopore sequencing machine. This is the smallest one they produce. And so I'm going to pass this around. If you drop it, don't worry, this is a broken one. So for circa £1,000, you can sell your soul to the devil and get one of those. You can take it to your lab and you can start using it. Because it doesn't matter to me what technology is perhaps the best one to pick out of this toolkit for doing it. The best sequencer you have is the one that's on your bench in front of you that you have access to. You know, if you want to do something really fancy, then talk to somebody involved in genomics, but this is a really enabling and disruptive piece of technology. There's lots of false propaganda about how accurate and how inaccurate the nanopore is. Don't worry about it. As I said, you know, think about the application that you're going to use it for. If you have to rely on it in court, I suggest you don't use the nanopore. Otherwise, if you're careful, you can use it very, very, very effectively. And I'll come on to how to do that later. But I think it's really important to say this technology is getting better all the time. There are little tweaks. And the PacBio also isn't very accurate, but one of the reasons why it's more accurate is that we look at that molecule again and again and again, and we can build a more accurate picture of what's going on. So starting to be able to do that for the nanopore as well. So I think what I want you to kind of take away from this is that different technologies do different things. They're applied better in certain situations. If you're thinking about surveillance versus diagnostic, perhaps one of these is more useful to you. It depends also on the system. If it's really complex, perhaps you need to generate a lot of data to understand that sample. But as I said, it doesn't matter which one of these you've got. Even if you've got an ion torrent in your lab, you can still use it for doing genomics. So all genomics approaches have one thing in common at the moment, which is that we need to take a sample and render it into a form that will fit on the sequencer. And so one of the reasons I put this in here is that we're looking at DNA, RNA. In most cases, what we're doing is we're changing that RNA to DNA so we can sequence it. These are DNA sequencers. The exception to that is the Oxford Nanopore technology, which can sequence native RNA. And I suggest that you don't do that unless you really have to at the moment, because that's an emerging piece of technology and usage. But we can also, within these different workflows, as we change the DNA or the RNA so that it's compatible with the machine, we can also do things like target particular types of sequences or particular aspects of that raw nucleic acid that we're putting onto the machine. So by targeting, what we're doing is we're selecting, and most people are doing PCR or they're doing some sort of hybridization, matching two pieces of DNA together so they can be pulled down and sequenced. Now, the reason I talk about this being an end-to-end process is that how you make that library impacts on how you make that library. In other words, how you change that DNA so that it goes on the machine has a big impact on how you can do the analysis and what you can expect to see in that. Yeah? As a really obvious example, don't expect to be able to see an RNA virus genome if you've prepped it for DNA. Yeah? It's obvious, isn't it? But there's lots of little mistakes like that we all make, and we can remove things from our sample before we've got anywhere near the sequencer. And the way that we change that DNA, the way that we sequence it, means that we've got different then approaches for looking at that analysis. We can look for things that are unique and novel. We can look for things that look like something else. And we can also then look at those pieces of DNA by themselves to get some sort of overview. So each one of these processes, again, has to be thought about ahead of time so that you know how you're going to do your analysis, how you're going to get the most out of your sample, how you're going to find that novel virus in the sample that you're looking at. So one of the things that has really allowed us to use this sequencer that's been passed around at the moment is that a group of colleagues got together and thought hard about how to produce an end-to-end system to generate real-time epidemiological information. And they started thinking about this in the context of Ebola and influenza and then eventually SARS-CoV-2. And what they realized is that they needed to have an extremely rapid turnaround, a real-time approach that provided actionable insights from the data from patients. And so they developed what they called the Arctic network. And basically what they did is that they designed a process by which you could make lots of little PCRs along a larger genome and then use those to reconstruct the genome and look at that. And this is how most of the SARS-CoV-2 genomes were sequenced during the last pandemic. So my experience, as I said, was being involved in COG, where we had 16 regional sequencing centers and the Sanger Center as a large sequencing site. We were involved with the 4 public health agencies, and we were funded by a large array of funders at that time. And this project was put together by Sharon Peacock and was really driven forward by her. We had some very clear objectives that had been mangled in the conversion to PowerPoint. So, but basically it was to generate the data in real time. It was to make it available as quickly as possible and be able to support the national and international collaborations to understand this virus and to track outbreaks. So we were able to use existing sequencing sites because there'd been a lot of investment in genomics in the UK, and we were able to use some infrastructure that had been previously funded, and I'll describe that in a moment. But we had 2 basic streams. We had one coming from healthcare, and we had one coming from the PCR testing. As a general breakdown, the Sanger did the community PCR testing, and the smaller hubs did the NHS samples and care homes. And at various points, there was a bit of ebb and flow between those. What was important was that all of this data went onto a trusted research environment, MRC-funded cloud server, which allowed all the data to be put there and looked at to be interacted with by the health agencies. And then we put together reports with the health agencies, and that reported to government. It was fed back to the NHS labs and was then also used for ongoing research. So this is a quick map of the country and just breaking down where those different sites in stars were serving the communities around them. And one of the things that I want to tell you, because I think this is a group that's going to be thinking about doing this sort of thing, this was really a rapid stand-up, and we got sequencing very, very early after we got the right funding in place. And by April, we were able to show early proofs of concept that this was going to work and that it could be useful. And that meant that we were in the position to support a number of research endeavors, looking at the severity of disease, immunity, transmission in hospitals and care homes, and also to support evidence for community testing, supporting some of the big sort of funded studies such as ONS and REACT. So COG was able to stand up something very quickly. We sequenced on average about 10% of cases in the UK. We developed the CLIMB COVID computer analysis network, which integrated everything centrally. We did the QC of the data and the analysis in a standardized way. We made that data available and open access to share with the UK researchers and internationally. And we were able to share our findings and our methodologies across the world, leading to some training international networks. There were some key scientific findings about tracking of importations in the early stages, the identification of mutations in spike proteins, the detection and characterization of the Kent variant, and the cancellation of Christmas, and the use of genomics for, well, yeah, it was, lots of people take the blame for it, but that's the, the cancellation of Christmas and things like that, that stemmed from an understanding of how the virus was spreading and what was going on. So the use of genomics in outbreaks as well was also found to be useful, but one of the things I would stress is that the good epidemiology actually quite often told us what was going on, and the genomics confirmed that. It wasn't like that genomics was always breaking new ground in this, but it was giving that backup and that assurance of what was happening. So in terms of surveillance, we were able to produce these lovely data sets of showing how the variants were changing during the pandemic, and that graph is shown here with the different colors being different strains of virus and how they were changing over time. As I said, that was only possible because we had an idea of how to do it already, because the Arctic Network had been funded and was ready to respond with a design for SARS-CoV-2. And that also we had that data infrastructure in place through the CLIMB Big Data Network. Again, funded, actually that funding got by Nick Lohman, and who was involved in the original Arctic bid. So we had some, as I said, good data. We knew about the coverage of variants and how many we were sequencing in each location within the UK. But for me, coming back and looking at this, I think what we kind of really began to understand is that the main barrier to actually responding to these situations genomically was actually access to samples. I spent a lot of my time leading a genomics endeavor at Liverpool by phoning round hospital labs, asking them, can they please send us this sample? Or listening to a minister asking us if we could sequence more more viral genomes than we currently were. And most of the problems there were to do with access to sample. It wasn't our ability to keep up with the tidal wave of infections. It was actually getting hold of those and processing them that was the main point. And I think one of the things that you can't see very well on this, but this pyramid is basically showing NHS, allied universities, and then at the bottom, centralized sequencing facilities, this pyramid. And the reason why I'm showing this pyramid is up at the top, what we've got is we've got low throughput, what we've got is low throughput, and then at the bottom we've got high throughput. So we can have these sorts of different levels of understanding. But with the low throughput, we can have a really fast turnaround time. But with the higher throughput, say at Sanger, what we found is actually a lot of that data was delayed because that high throughput demanded a longer turnaround time. And I've added this yellow arrow down the side here, which says surveillance down it. It was very visible on my Mac, but it says surveillance down it. And what I'm really just saying as well is that in some ways what we do is we move from diagnostics to surveillance down this corridor. Diagnostics being quick and low throughput perhaps, through to, when we get to the higher throughput and the low turnaround time, this is more about surveillance. And the reason I say that is because when we get to that point, we're no longer able to use that data to inform patient care. Now, in the case of these genomes in COVID, we were not using these genomes to infer patient care. And I think that that's a really important thing to do, because if we were, we needed to be closer to the top of this pyramid. There's lots of problems with this sort of surveillance as well, and many of them are actually solved using wastewater sequencing, because that allows us to get an unbiased approach. We can detect things early in the wastewater system before we see those things in the hospitals, and it's also very cost effective. We went on to use this with Julian and others to look at minor variants in the data, which again this is very good for, and to say that basically the surveillance and so what I wanted to say sort of in summary of this part was that whole genomes are capable of providing really accurate information. And I just want to, how long have I got? 5, okay. What I wanted to say is that what we've done since then is take the sequencing into the diagnostics and try and work really hard on getting things to be quicker. So we've got one example of using 16S in the hospital locally, where we've put one of those nanopore machines into the lab there. And the result of that 16S test, sequencing just an amplicon, is that they've reduced the amount of antibiotics that they're using, and it's having a big effect on antimicrobial stewardship. We're currently developing this for a number of viruses, but Alan gave you the example of the coronavirus, and I just wanted, in dogs, and I just wanted to say that the diagnostic was failing actually for this coronavirus because it wasn't giving a very good readout in the phylogeny. And the implementation of whole genome sequencing allowed us to fill that gap in the phylogeny. We're currently doing this for HIV and for norovirus, which is a lot more difficult. So, what I wanted to end with was that one of the things that we're trying to do a lot at the moment is use metagenomics to simplify the type of analysis that we're doing. And what we're finding is metatranscriptomics gives us everything. Sequencing everything in a sample of DNA is giving us really good bacterial genomes. And we're doing this with the PacBio because it's such an excellent high-quality sequencer that we're able to get out of stool 64 complete genomes of bacteria from a sample in one go. So what I think is we're approaching this sort of period where genomics can be used realistically. Pathogens of interest can be identified from complex samples such as stool without the need for isolation. And RNA provides us with a one-hit analysis where we can look at viruses, bacteria, and also parasites in one sample. So I just wanted to acknowledge one of my funders here, and then to acknowledge all the people that I've been collaborating with. Thank you very much. The problem with this room is it makes you very laid back. I don't know if anybody's noticed that.
I think it's the leisurely lunch as well. It just kind of slows the pace down. Yeah. Do we have any questions? I think I saw this hand first, and then I'll come to you, and then I'll...
Because this is a CAT conference, and I'm going to ask you something just to expand. There was something very odd that happened in wastewater in Liverpool, and then I think in Newcastle, and we think it was adaptation for rats. That was an unexplained finding, but it was really quite exciting. Could you just go into that? Because I think it demonstrates the power of your technology.
Yeah, so I think what's interesting about that as an example, so what was turning up in the wastewater sampling was unexplained variants which didn't seem to be in the human population. I think it's important to say we also found variation in human virus that wasn't seen elsewhere in the country. So I think that there's a story around these coronaviruses being a lot more diverse maybe than we thought. And, that we hit on these small examples where, there was a local expansion in a particular genotype that then seemed to go extinct or was never surveilled actually within the human population. Because we were only looking at 10% of human cases in those samples. So again, that's where the wastewater gives us that extra capacity to look for different sequences, but then also has the ability to throw some red herrings maybe along the way. And so it sort of shows you that you can't rely on one type of surveillance to get the full picture, I think. Does that answer that?
Thank you. We have one question here, one at the back, and then one in front of that. Thank you.
Thank you very much for that talk. It brought me up to date from my postdoc days when we used to do cloning sequencing, which was very time-consuming.
Don't do Sanger sequencing
yes,
anymore.
Exactly. But the question I have is, we're down to single molecule amplification. If you extract either DNA or RNA from a fecal sample, for example, there would be a mixture of organisms there. How do you differentiate between, do you use different primers or a specific primer?
Well, so I think, again, so that allows me just to explain something I didn't have time to explain. Thank you. So metagenomics, or looking at microbiomes, is exactly that. You embrace the diversity of a sample. And so there's a couple of ways you can do that. Is you can just look at all of that diversity and hope that you've got enough of what you're interested in to actually describe it. Or if you know what you're interested in, then you can go into that complex sample with a specific method for enriching for it, or for getting those particular types of genome. So one of the things with the tiling arrays with the Arctic protocol is we had to keep on changing that during COVID because it was so tied into the genomes that we were looking for that as the virus evolved, we had to change it to keep up with those changes in variants that we were seeing. But you don't need a whole genome to do surveillance, okay? Bits, you don't need the perfect whole genome to do that.
Thank you so much for your presentation, first of all. Going back to how you mentioned AI can be used in genomics, do you see any potential issues in the future in terms of accuracy with the use of AI for whole genome sequencing?
Yes. Thank you. I think one of the things that we're most concerned about at the moment in the community is basically issues around previous biases in data sets, and so it's, there's been a lot of bias in the type of data that we've collected, so if you take the human genome, for example, that's a white human male that was sequenced first off, and it's taking us a long time to produce a pan-genome that is representative of, race and diversity that we expect to be there. So, for example, if an AI model was trained on just one example of a genome, then there's a really good chance that it won't pick up other things that are necessary or would be thought to be there. So I think a lot of the problems that we see potentially at the moment are around the training data and any inherent biases that are there.
Thank you.
Yeah, thanks. I was going to mention the rats, because we got involved with that as well because of the animal health part to that, and brought in Mark Viney, who was doing quite a lot of work at the time on picking up rats. One difficulty was that we thought maybe we could get students to go down into the sewers and collect rats for us, but I was told health and safety wouldn't let me. But actually, that was the difficulty, is getting the good rat samples, because by the time you're sequencing downstream, it's been diluted so much. And we'd seen the New York papers about rats and so on, but actually there's probably so much cat and dog DNA in the water as well that you can't even necessarily...
I didn't think it was necessarily my result to talk about, but I don't think we actually found it directly in rats in the end, is that?
That's right, yeah. There was some serology that was interesting, but not from that Liverpool site. I think it did eventually go back to a human that had an unusual variant. But also, because I think all of this work is just fantastic, and we've now got a big research program looking at how we use the new technology. We have to be really careful from the animal health side when you're looking at notifiable diseases because of the trade impacts that it can have. So I was interested to hear you say, don't use it in court, because that's where we'll end up with a judicial review.
I mean,
if
it
we
wasn't
start
a flippant remark.
No, it's absolutely correct.
And also been asked to comment multiple times on things like that, and I've decided that that's not my expertise.
Damn it! I was hoping you'd give me the answer.
Well, I think the thing is that there is also a really interesting problem. If you look for something, you find it. And I'd be happy to talk to you privately about a few of those examples if you're interested. Yeah.